Well, I'd like to welcome everybody to this webinar today, Grapevine Red Blotch Disease, What You Need to Know. My name's Frank Salem. I'm from University of California, Davis in the Department of Entomology and Nematology, and I'll be moderating the session today. What we have is about a number of presenters from different laboratories across the US and Canada and in Switzerland that have been working recently on red blotch. We have over 400 registrants for this webinar, so I think that speaks to the significance of the issue to a number of different people, and we're hoping to provide you today with a lot of new information on red blotch that are coming out of a lot of these different research groups. And we'll have an opportunity at the end of the webinar to address questions. So we'll have a question and answer session where you can enter your, after Dr. Galino's presentation, we'll have an opportunity for you to start entering questions, and a number of the people on our panel then will be able to address those particular questions. We're also going to ask you to complete a survey. We'll give you the link to that survey later in the program. It's just a very brief survey, less than 5 minutes. That will give us some information on how you like this webinar format. Also, what it is that you would like to know more about red blotch, and hopefully we'll be able to get that information together and out to you, because again, we have a number of different speakers here that have very detailed expertise to this point on the disease. With that, I would like to introduce our first speaker, who's Dr. Mysore Sudarshana, and he is a research scientist with USDA-ARS, affiliated with the Department of Plant Pathology here at UC Davis. He's been with the ARS group since 2008. He's been working on the biology, detection, and management of these viruses affecting grapevines, stone fruits, and walnuts, and he'll be talking about some of his research on red blotch today. Sudi?
All right, thanks, Frank. In 2008, when I started my job, I was focusing on the grapevine, the trunk disorders, mainly the union disorders. And at the time, we were looking at some of the Pinot-Ventnor interaction problems, and also visiting some of the Syrah decline problems too. In one of our visits to a vineyard in Sonoma County, we went there to look at the Syrah disorder, but I noticed a Malbec planting in the vineyard showing the symptoms here, kind of reddish leaves, but not typical leaf roll. I was kind of novice to leaf roll and everything at that point. But it looked kind of distinct from the what you see in grapevine virus books. It's not like typical leaf roll. Then the same day afternoon, we went to another vineyard where again they had told me about some problem there. This is Cabernet Sauvignon 337 clone in Napa County. Again, here also you see the leaf blades were flat and they had blotches. And again, these were also not like typical leaf roll, although we kind of suspected it could be leaf roll. At that time, Dr. Rohani and Dr. Emoto, Dr. Rohani is from FPS UC Davis. Dr. Emoto was my predecessor in USDA, worked on for several decades on stone fruit and grapevine viruses. They kind of thought it could be leaf roll, and when we tested, it didn't have any leaf roll. So moving on, at Oakville Station, Jason Benz was the station manager at Oakville Enology and Viticulture Research Station of UC Davis, and Dr. Jim Wolpert and Mike Anderson, working with Jim, they had noticed around the time a kind of red blotch kind of symptoms, and those wines also had tested negative, and they had sourced the material from a very good source. So they were wondering what was happening. And in 2009 summer, they asked me whether I can take a look at it. It could be another leaf roll strain or a new leaf roll virus, and they asked me to come and look at it. And then when we went there, we found actually it's not just one clone of Cabernet Sauvignon in station, but in nearby vineyards we had Cabernet Franc and several Scions on different rootstocks. They all had this problem where they tested negative for leaf roll viruses, and they had a problem with blotch symptoms. This is what we saw at the time on Cabernet Franc leaves in a flat leaf blade and blotchy symptoms. And characteristic thing, when you turn the leaf on the other side, you can see on the lower side the reddish pink veins. So we thought it's not normal leaf roll or anything that we see of. Okay, so we designated this disease as grapevine red blotch, and went about looking at what was happening here. And I collaborated with Dr. Adip's group. Maher helped us in some of the work, and Ashita, my technician in my lab at the time. So we made extracted double-stranded RNA, looked at cDNA libraries by next generation sequencing, and then when we analyzed, what we found was a novel DNA virus at that point, and we immediately developed primers for detection. And when we started working on this, we didn't even know. Actually, we had 3 isolates, and 2 happened to belong to one strain, and one isolate was a different strain, which we called them as 2 clades. And Keith is going to talk about that. So immediately we went around and looked at more vineyards, and we saw in 6 commercial vineyards around the station, this virus was present by PCR tests. And so we were holding on to get a little more biological information on the virus. And then in early 2012, Dr. Kit Perry from Cornell contacted us, and he told us that they have a new virus, whether we know anything about it. And we shared the information, and we came to know it was the same virus. So then it was a question of associating virus with symptomatic wines, and then we were able to do that. And then end of 2012, we disseminated the information to scientists and other stakeholders, both on the disease and the virus, everything. Okay. And later we came to know that an identical virus had been found in Washington State, and commercial labs started reporting this virus was present in other states in the US and Canada. So when it comes to red leaf diseases, one problem we have is symptom-based recognition of virus or non-virus, etc. So we can have 2 kinds of factors, abiotic and biotic factors, that can cause red leaf diseases. Abiotic factors would mean like tie tape or injury to trunk and corons in vineyards, and sometimes the nutritional problems. When it comes to biotic, we can have crown gall, Graves' disease, cankers by fungi, and incompatibility reactions with when we have latent viruses, then leaf roll disease also. Here is an example of a tie tape issue where a coron is constricted and we get reddening of the leaves. And here also a tie tape problem. This part of the coron is showing red leaves, but not this part of the coron. And then potassium deficiency. This was one of the reasons, actually, a lot of people who had noticed red blotch symptoms, they thought it was a nutritional problem, and we came to several vineyards actually were trying to amend or ameliorate the symptoms by application of potassium. Okay. When it comes to biotic factors, we have here's a crown gall in a new planting, which is the gall has caused constriction, and you see the red leaves, and canker here, and incompatibility reaction due to a latent virus, which we don't know yet, this one, and what this virus can do if it's in cyan or that agent. The rootstock can react to that agent, and we get incompatibility or graft union, necrotic, turning necrotic, and resulting in red canopy. And here is leaf roll. Most of you have seen this. That's a classic leaf roll symptom. So now bringing back to this, so where would red blotch fit in? So here is a Cabernet Franc wine having red blotch, and here is a Cabernet Franc wine having leaf roll. You can see the leaf blades in case of leaf roll, cupping rolling down, and here you can see the reddish veins in red blotch, but we don't see that here. Going to now looking at symptoms in different cultivars. This on Cabernet Franc. This is on a second leaf Cabernet Sauvignon. Nearly 95% of the vines were infected at this point. And a year later, you can see this. Cabernet Sauvignon shows kind of a little milder symptoms compared to Cabernet Franc. This is Malbec. You can see almost entire blade turning red. Usually, always the symptom starts early fall or late summer to early fall on basal leaves, and gradually the terminal leaves also show symptoms. And here is in Merlot. If I had not seen red blotch before, I would have easily pushed this off as some nutritional problem. And this in Petit Verdot makes a nice greeting card, and you can see terminal leaf showing color, and there's red color. What's important is, in all these cases, we had tested, didn't have any leaf fold viruses, only red blotch at that point. And some of them, of course, had persistent breeding virus, which we don't consider as important in terms of symptom traction. This is in case of Pinot Noir. And the worst thing we have seen is in kind of Zinfandel. Here are two self-rooted Zinfandel. This in Mendocino County. And you can see the leaves turning crispy. And they also had red blotch symptoms, but the leaves were a lot more sensitive for the virus as the summer progressed. And here is in foothills in Amador County, thanks to Lynn Wunderlich. In case of white varieties, symptoms are not easy to recognize. And here in case of Chardonnay, Rwanda is very good at it. And in white Riesling here, I can't see this as a virus symptom. And in the Zan, thanks to Mark Bettany. And it's not like always straightforward, whole canopy turns red or anything like that. You can have one part of the cardon virus-free, the other cardon virus-positive, and having red blotch symptoms. And some of these vines had been tested for several years, and in 2014, we saw some new infections starting just on single shoots. The disease gradient, Brian is going to talk about it later, can be, it's like a moving edge, or it can be random, depending on whether there's a vector or not. So what is the significance of red blotch to grape industry? Okay, the first problem. When you have red blotch in a vineyard, there's a delay in maturity. The bricks will take a little longer time to reach the level required for harvesting. So here is a vineyard where the healthy or normal wines had been picked already. Those with red blotch were still having the clusters hanging. You can see here and also here. And when you have a second leaf vineyard showing red blotch like this, it's totally unproductive. And this vineyard was pulled out after fourth leaf, 4 years later after it was planted. And a winery which worked with me, they looked at those vines, and after we tested by PCR for the virus, they looked at the bricks and titratable acidity, pH, anthocyanin, etc. In 2012, they could see a difference of about 5 units reduced by when you have red blotch, and acidity was also higher, and anthocyanin was reduced. And in the following year, the difference was a little less noticeable for bricks, but still anthocyanin content was very much reduced. The revenue forecast they had in this scenario was, if you had $100 per bottle, no red blotch, you're looking at $90,000 per acre. When you have red blotch, this wine has to go for bulk, and the revenue would be $10,800. That's totally undesirable. And this was the problem. A lot of vineyards producing premium wine in Northwest and other vineyards, other places for Facebook. Several of the vineyards we looked at early on, about 11. We did not tell them. It was just the vineyard owners, the managers. They made a voluntary decision that it was not productive to keep them. And once the virus presence was confirmed, they were all removed in these years. And actually, all those vineyards I worked on in 2011, 2012, we don't have them anymore. This is the only one there, and this other one. So, to summarize, red blotch disease is a recently recognized disease of wine grapes. The virus now known to be present in most of the grape production regions of the US and also in Canada. Symptoms can vary considerably among grapevine cultivars, so it takes a lab test to confirm the presence of the virus. A PCR and qPCR tests both are available for detection of red blotch virus. And the primary effect of the disease is on total soluble solid accumulation and also volatiles. And of course, in Zinfandel, we are seeing now in the last 2 years that it can affect the prune weight. Some of those things have not been observed carefully, but we see the disease having a damaging effect on Zinfandel vines. Well, just like a Hollywood list of acknowledgement at the end, but I think from 2008 now it's 16, and I haven't even finished 8 years yet. And if getting this virus and the disease to be recognized as an important problem, I needed collaboration from lots and lots of people at different points. So I don't want to name everybody. Here's a list. And most importantly, I thank all the funding agencies. And recently we got the Sierra Black grant to move on to the vector, finding the vector for red blotch virus. And several growers and nurseries also cooperated with me. They were proactive in getting some of these things addressed. Thank you, Frank. I think this should conclude my part of the presentation.